Hello, good afternoon. Good afternoon, everyone. Good afternoon, participants.
Good afternoon.
Good afternoon. Yes, Professor, nice to see you here today.
Thank you.
Okay, today is day 3 for our training, and so far we are doing good. So want to just continue to thank the participants. They always try to join on time. I see the number is increasing. And in the interest of time, we have to start exactly at 2 as usual. So you are most welcome to day 3 of the training. And we ask your participation and concentration during the training. So we have Prof. Rogerio in the meeting with us today, so he can give a word or two before we start with our meeting. Prof, you are welcome. And your camera seems to be closed. This, I think you need to check the camera. Yes, now we can see you.
Thank you very much, Selma. Good afternoon, particular to the facilitators that everything they've been doing to make sure we have this wonderful training on advanced statistics and experimental designs. Good afternoon to Ruforo and the Secretariat. Welcome to this 30th day of session of training in advanced statistics and experimental design. One thing that I would like to ask you regarding this training is to make sure you put your contacts in terms of email and telephone number. The World Bank has started requesting us to submit the list of participants of the first training that we organized, which is a R training, and then the second on proposal writing. So please provide us your email and your contact numbers, because we need that information to provide to the World Bank. The World Bank in 3 weeks' time will start contacting you. So be aware that the center is organizing this training, the Center of Excellence in Agri-Food Systems and Nutrition of Eduardo Mondlane University. And with that, I would like to invite you to enjoy this training and take advantage of this for the good of all Africa. Thank you very much.
Thank you very much, Professor. I think let me also assure the participants at the beginning of this training that we do notice, and we are aware that there are some of you who have not received certificates for the previous trainings. So we are going to reconcile all the list and ensure that we check out for the names which were left out and give the certificates. So please be assured that the certificates will come. Professor, we shall provide the list of the contacts. We've been collecting their contact details on the forms. We just need to reconcile and send them to you. So please do not put your names in the chat box. The registration link is going to be shared. You register as usual. Make sure that you register. And also, please do not put your questions in the chat box. Otherwise, the facilitators will not be able to see the questions. Put your questions in Q&A. So with that, thank you very much for joining, and thank you to the facilitators. Professor Susan, I hand over to you and your team. Thank you.
Very much. Can you hear me?
Yes, we can hear you well.
Okay, thank you very much, Selma and Professor Rogerio. We are happy and excited to come to day 3 of our training. Would like to thank the participants for logging in a large number. Would like also to extend our thanks to Food and Nutrition Institute for organizing this training and funding. Also thank World Bank for funding the training. We would like also to appreciate RUForum for organizing, providing us with space, internet, and the webinar to conduct the training. I will hand over to Dr. Thomas to complete what he has prepared for you. And then after Dr. Thomas, we will change and go on to something else. Thank you very much. Over to you, Dr. Thomas.
Hello, good afternoon, everybody. You're welcome to the third day of our training. The time is always not our best ally, but we would rather cover something little and people go with knowledge rather than covering everything and then leave most people without. So I'm going to give brief overview of experimental design, and then there are a few data sets that is already provided to you with the scripts. So some of this we are going to ask you to analyze yourself, and then we can follow from there. So let me just give my overview, probably 1 hour or 2 hours, and then I give my colleagues to continue. Okay, so an experiment we know is the only way to establish cost-effect relationship. When we are talking about experimental design, there are basic principles that cut across. It doesn't matter which field you are, so everybody should feel comfortable here, whether you're doing agriculture, biology, ecology, social science, economics, and so on. The only thing here is that sometimes we give example, we give example that really to illustrate the point, and then you can try to put it in perspective what your situation is. Of course, those who have gone to the field, they know the way experiments are laid out in the field. So from here, of course, you see there's the issue of shading. There is also some kind of a patch that seems not to be not really to be doing very well. So these are issues that needs to be taken into consideration when designing an experiment. You also have experiment in the lab. Those who do food science, those who do chemistry, those who do technology, engineering, and so on. They do also a number of experiments in the lab. So things like test tubes, petri dishes, these are a lot more uniform, and that has implication as far as doing your experiment is concerned. So when we are talking about experimental design, there are basically two things that comes to mind. The first thing is the issue of the experimental material, or what are the, you could look at it in terms of the type, in terms of the nature of your experiment. So this is where variation comes in. If you're an animal scientist, animals may be your experimental material. If you are a social scientist, a human beings. My screen not showing? Okay. Okay. Sorry, I was told my screen is not showing. Let's hold on a bit. Well, let me first sort my screen and then I get back. Yeah, so I'm going to share my screen, but I'll continue talking. that. No, I don't use Windows, I use a Mac. So, so all the Mac products are genuine. So then, I guess, yeah. No, I'm trying to. I don't know what, but I don't want to have it here. View, can I have it as a full screen? Okay, let me try to share the diagram. Wait a bit. I want to first go to the first page and then let's see there. Can you check, confirm?
Yeah.
Okay, great. Okay, sorry for that. Let's keep monitoring to ensure that I'm showing you something that is authentic. Okay, so we have can do experiment in the lab. So my point I was trying to emphasize here is it depends on the field you are. We always use different experimental material. If you are in agriculture, like crop, mainly the experimental material that I use are mainly land. You can use pots, you can use basins and any other. If you're an animal scientist, you can use animals like the way we're seeing here. Different animals may constitute your experimental material. And then if you're a social scientist, you may use human being as your experimental material. And then the other thing is the treatment set. So these are what you're trying to test the effect. So here, you can control the type of variation that is caused by the treatment. So if you look at my picture here, I have what I call treatment on this side, and then have the experimental material on this side. So if you're dealing with animal drugs, for example, then you need the animals are supposed to be your experimental material. So for a pot experiment, for example, you probably this would do pot experiment. The pot here with the soil will constitute our experimental unit, and then the plants, the maize plant here could be. Then you could be having things like fertilizer or pesticides or variety, and that could be your treatment. So the treatment has to be defined very, very clearly. If your treatment is not well defined, then you're not able to go to do any data analysis. Remember, when you're doing analysis of variance, we looked at the issue of what to convert factors and that. So that needs to be very, very clear. And you also have to be very clear what you're measuring. Are you measuring plant height? Are you measuring number of copes? Are you measuring leaf length? So this also have to be very, very clearly. Sometimes you find that somebody's writing a thesis, but they cannot differentiate between what an experiment, what is a treatment, and what are the variables that are being measured. Sorry, I apologize for these pictures. If you get offended, sorry, this is just to illustrate my point. So there are basically two things that an experiment should give you. A good experiment should be able to estimate the differences between the treatment that you're comparing. So here we have I have two toy examples here. I'm testing 2 energy drinks. So these are my experiment, my treatments, and then the human beings in this case are my experimental unit. So in the first case, for example, we can see that there is variation in our experimental material. So from here, you can see that we have a small guy fighting with a big guy. So that may pose a bit of challenge here. And then here you have experimental units that are kind of similar. So the whole idea behind designing an experiment is that you want to design your experiment in such a way that you have an unbiased estimates of the differences between the treatment. So what could be wrong with the first case? Can somebody in the chat tell us what could be the issue with this? In experiment 1, what could be? Okay, somebody said there's going to be some biasness, unbalanced. Okay. Yeah, so there's certainly going to be bias. Bias is that one of the treatment is going to be favored and the other one is going to be unfavored. So if, for example, this young man here somehow succeeds in putting the big man down, we will get a bit confused whether it is because he took Red Bull or because he's smaller and he's able to move quickly and dodge the big man. So that's the kind of thing. So here the interest is that. So we need to compare the effect of the two energy drinks. We need to have unbiased estimates of the differences. A good experiment should also provide for us what we call unbiased estimate of experimental error variance. So here, for example, let's say we have 4 rats, this is our experimental unit, and we give them the same treatment. Now, you certainly, you're not going to get the uniform reading. There are going to be some slight variation here. Now, this variation between similar experimental unit receiving the same treatment is what we call our experimental error. So experimental error variance is very important because when you're doing any statistical test, you need the estimates of experimental variance in order to calculate statistics such as T statistics or F statistics. So that becomes very, very important. Now let's look at the second case now. Now in this case, the treatments are all the same. And then you can see here, we have a lot more variability. So this is time taken, maybe let's say whatever it is. So you can see from here that the smaller dogs taking, they have a score of 50. The bigger dogs have a score of 500. Remember, they're all receiving the same treatment. So when you are designing your experiment and you make a wrong assumption about the experimental unit, it's likely to cause a lot of noise or a lot of variability in your analysis, and you may not be able to identify, you may not be able to detect the differences between the treatment that you have here. So in this case, we cannot assume that all the variation here are due to experimental error. There's also variation due to the differences between the different animals. So basically, I know this is a review, it's kind of a repetition for most of you. Basically, there are 3 principles of experimental design. One is replication. The second one is called blocking. And sometimes it's called local control. Sometimes it's called restriction. And then we have what called randomization. So what is replication? So replication is simply repeating a treatment under identical condition. So each treatment is applied independently to 2 or more experimental units. So here, for example, we have a situation where each rat receives the same treatment, but independent of each other. So each of them will constitute a replicate. But on the left-hand side, the rats are receiving treatment not independently. They're receiving it together. So the whole of this is actually an experimental unit. Then each rat becomes a sampling unit. And if you decide to take each rat as a replicate, this is what we call a pseudo-replication, which is not true replication. So you need to be aware of the pseudo-replication. So why is replication important? Without replication, you will not be able to estimate the error variance. So if we have this 10, 9, 11, and 9, so you can run this in error and you get, so for this group here, the variability is 0.72 or 73. Now for the case of the dogs, the variability is up to 42,358. So you can see there's a lot more variability here. Now it would be extremely naive to consider all this variation to be experimental error because this is an overinflated experimental error. So this effect of the animal size is also included in the variability that we have measured there. Also, we all know that replication increases precisions of the parameter you're trying to estimate. So let's say, for example, you're interested in estimating the time of reaction of a rat to a poison. I said the rat is given a poison, and then it takes maybe, let's say if you have a smaller experiment with 8 rats, if you have a bigger experiment with 8 rats, the variance will be 0.78. Now, if you look at the variance of the mean, is going to be the variance that you calculated divided by the total, the number of replications. So you can see with 8 rats, the variance is, sorry. So you can see with 8 rats, the variability, with 8 rats, the variability is much smaller than if you're dealing with only 4 rats. So the smaller the variability of the estimate, the more precise the estimated result. So you can see with 8 rats, you get higher variability, lower variability compared to 4 rats. Other roles of replication include avoid losing the whole experiment. So here, for example, if you have your experiment somewhere in the village on the farmer's field, there may be goats running around. So if you have only one plot, the goats might actually eat up the experiment, one of the plots. So if you have 5 plots, they can eat 2, and then you have 3. So that will still be able to save you as a student. Replication also increases the range of validity of the results. So here you observe results of a wider sample space, maybe soil, maybe farm, and so on and so forth. Yeah, this one always reminds me of a story of an old lady and an old gentleman in my village who decided one time that the old lady claimed that she can actually wrestle the man down. The man said, no, that's not possible. So they were arguing from the house. So they say, why not? We are all here, let's try. So the woman puts the man down. The man said, no, you know your house very well. Let's try, go outside. So when they move on one side of the house, the man was put down again. When they move under the tree, the man was also put down again. They move a little bit to the neighbor's garden, the man was also put down. So under this, we have validated our result of a wide range of situations, and we are confident that whatever result that has come out is actually valid. I talked about pseudo-replication already. For those who work with samples, you need to be aware of what we call biological versus technical replication. So biological replications are like from leaf, one leaf. If you get one leaf and extract one sample from that leaf and test that, that will be one biological. But you can extract 2 samples from the same leaf and you have 2 replicate like A1, A2. So A1, A2 in this case is what we call technical replication. Those who run experiment in the lab, they know that when you're doing soil analysis, for example, you may need to, from the same sample, you may need to run 3 or more replicates. So those are more called technical replicate compared to the biological replicate. So you need to be aware of this. The technical replicates are just like pseudo replication that we are talking about. Just briefly, always I know in agricultural experimentation, always people say, I've used 3 replication. So 3 replication has become like a kind of a trademark for anybody who is doing experiment in agriculture. And if you ask people why, no one can even explain. Yeah, but the number of replication will depend on one, how much variability is there in your experimental material. So if you look at the at the animals on the left-hand side, and then you compare with animals on the right-hand side, which of the animals, which one would you consider to be easy to represent? The one on the left or the right? Which group of animals would be easy to represent? Okay, the left. Okay. Why the left? Okay. So the animals on the left are more uniform. I hope your left is the same as my left. So the animals on the left are more uniform. So it's easy to pick one animal to represent them. So if you're dealing with material as variable as the one on the right, that means you may need to have a lot more replication in order to be able to represent that group fairly well. My colleague Ellen is going to talk about sampling at some point, and you'll be able to see how that one comes into play. The other thing is also the size of the difference that you want to detect. Now, if let's say I have these two examples here. Let's say we are interested, we want to set up an experiment to compare the noise made by a rat and the noise made by a lion. Do we really need to do a lot of replication? Do we even need to do an experiment? Okay. So you can see that the difference between the lion, the noise made by the lion and the noise made by a rat is so the difference is so big that you probably need to replicate it only one time. But when you're trying to compare the two rats of different colors, probably they make similar amount of noise. And in this case, if you have to detect this, you need a lot of replication. So for example, let's say you're comparing two new varieties of maize. Let's say you're comparing the old variety of maize with the new one that has come up. Now, the question that one would always want to answer is, how much increase in yield is acceptable? Because that you could set that as the margin that you want to detect. And if the difference that you want to detect is very, very small, that means you need a lot more replication. But if the difference that you want to detect is very big, then you don't need a lot of replication. Of course, also, how confident do you want to be? What is the level of significance? Yesterday, we talked briefly about, alpha, which is the what we call type 1 error. So if you want to be 99% confident, you may need a lot more replication compared to somebody who wants to be 90% confident. And then, of course, as a student or as a researcher, there's always the issue of resources. You may have the money, but then the experimental material itself may not be available. So that can also limit the number of replication that you can make. This material is going to be shared at some point, so don't worry. The other thing that I want to talk briefly is the blocking. So blocking, we are simply trying to control or minimize non-treatment variation in an experiment. So non-treatment variation can confound the effect of the treatment. Just like where we started from. Let me go back to this. So you can see from here that the non-treatment variation is, for example, the variation between the two gentlemen here who are wrestling. So if this young man somehow wrestled the big one, so you cannot tell whether it's because of his size or it's because of the energy drink that he has taken. So that's what we call confounding. So we always need to be very, very careful when we are designing our experiment. So non-treatment variation increases the noise in an experiment and makes it difficult to detect the treatment differences of any effect. So remember, we calculated this variation in this group and variation in that group, and we found that there's a lot more variability in this group compared to the first group. And part of the variation could be experimental error, but partly is due to the differences between the different animals. So we need to take the issue of the differences between the different animals into consideration when we are designing our experiment, so that we remove part of the variability that is due to the animal and only leave variation that is due to chance or experimental error variance. Okay, so here, for example, you can also see a situation where there's shedding of part of the experiment by these trees here. And then you can see that the soil that seems to be on the right-hand side of the trees seems to be more brown compared to the one that are on the left-hand side. So these are likely to have an effect on the treatment that you're going to compare. So at some point, you need then to come and look at your experimental area and try to take into consideration this variation that you see in the experimental material. And one way of handling such variation is through what we call experimental blocking in an experimental design. Yeah, of course. Let me try to check. Say the YouTube link is not working. Okay. So the non-treatment variation needs to be controlled. One way of controlling this is using homogeneous material. So what I need to do in that case is I can use only rats that are similar. So then I can avoid that. And then also the issue of management. Yes. In the chat, okay. Yes, there are standard formula for calculating number of replication that will take into consideration the things that I've talked about. Okay, yeah. So that one is there, but we cannot at this point go through all that because we don't have time. But the information is available. So there is the issue of homogeneous measurement, management. So things like weeding your experiment should be done uniformly. If you're going to take take a measurement, you need to take measurement in such a way that the people you're using to take your measurement don't introduce a lot of other variability in. And then there has to be also careful measurement. There's homogeneous management. But what we are going to talk about is what called blocking. So blocking can allow us to remove or control the effect of variation in the experimental material. So what is a block? In the simplest term, a block is a group of experimental units that are expected to be similar. So if we are doing an experiment with dogs, so we can group our dogs into groups that are similar. So for example, we can have block 1. With these dogs here, they look similar. We can go to block 2, we go to block 3, and then we go to block 4. So examples of blocks include plots with similar soil, same breed of animals, like what we are seeing in this particular case. So a block is simply a group of experimental units that are expected to be similar. Now, when we are talking about similarity in this case, the similarity has to be defined in relation to some characteristics. When you're dealing with soil, for example, the variation or the similarity may be in terms of soil fertility. In terms of animals, you may be interested in things like the initial weight, the weight of the animals, the breed of the animal, and so on and so forth. So this depends entirely on your experiment, what type of experiment you're trying to run. So in the field, we look at something like this. So you could, for example, consider this as each of these strips as a block, but it could also be 2 strips constituting a block. So here we assume that within the strip, there's uniformity, and this uniformity could be in terms of soil, in terms of water, in terms of shadings, and so on and so forth. Now, we always assume that the experimenter knows the experimental material which they're dealing with, and therefore they're able to actually group the experimental units such that there is actually some kind of uniformity within the group, and the group is what we call a block. And then the simpler of the three principles of experimental design is what we call randomization. So randomization is simply the process of assigning treatment to different experimental units using probability or chance. The emphasis here is that we want each treatment to have an equal chance of ending up in any of the experimental units that we are using in our experiment. So in medical studies, for example, maybe we have a new drug and we're comparing that new drug with some kind of placebo or even a standard drug that we have. So once we recruit patients who qualifies for our study, then we can take a coin and flip a coin. You say if the head comes up, you will be assigned to the treatment group. If the tail comes up, you will be assigned to the control group. And after that, you give them the treatments, and then you follow them up, and then you compare your results. So the good thing is we are going to see that ARA can generate for you, can randomize for you your treatment in the field very, very nicely, so that you don't need to worry about anything else. Okay, so in case of a plot, like a block, if this is what we have in the field, where we have block 1, block 2, and block 3. So here we can then do the randomization within each block. So the randomization process will be done within the block. I think it's really important that we do randomization because analysis, most of the statistical analysis that we do is based on the assumption that randomization has taken place. So if you don't do randomization, the validity of your result is not going to be okay. Your result is not going to be valid. I assume that you are interested in comparing the height of male and female. I mean, then someone say, oh, let me select tall male, tall female, male, and select short female and compare the height. So you can see from the start that there's no way you're going to come up with a valid result because you did not select your sample in a random manner. Okay, so those are the three principles of experimental design. I'm going to introduce you to 3 standard experimental design, and then I'm going to talk briefly about split plot and briefly about incomplete block design. Then I will try to show you some results, and we'll later ask you to try to generate those results yourself. Then maybe tomorrow morning you can ask one person to illustrate that for us. Okay. So the first design, the simplest of all the design that we use in experiment is what we call completely randomized design. And completely randomized design has one main assumption, is that the experimental units are homogeneous. So like you see, all these rats are similar, and therefore it doesn't really, I don't need to group them. Okay, so here the process of randomization will just happen at once. So you can randomize everything at once. I'm not going to, so here, this is just a simple way of generating, using ARA to generate for you, to randomize for you your treatment in a randomized complete block design. So let's say we are starting with this. So we need to label each of these as 1, 2, 3, up to 12. So you have the script is with you. So always try to read the comment. Like here we say, if we use this command set.seed, we set a number, say 500. Now this allows you to get the same randomization. So if you want to repeat the process, you get the same randomization process. Now, for now, I don't want you to worry that much about this. You can easily pick this script here and run it with you. So finally, what all this thing generated is that it generated for us a plan. So you can see rat number 1 will get treatment B, rat number 2 will get treatment C, number 3 will get treatment 9, like that. So this is the process. So this, as a result of assignment of the treatment, you can see that now we'll be able to assign our treatment to the different experimental units. And this assignment is done in R. What I'm trying to show here is kind of a complicated one because I was just trying to illustrate a point, but there is a much easier function in Package Agricole that can help you to generate either CRD or RCBD, as we are going to see. No. So completely randomized design, we assume that all experimental units are similar with respect to some characteristic of interest. So this is mainly used in the laboratory, in greenhouses, and application in the field is very, very rare, although sometimes it's possible to do it in the field. So why CRD cannot be used very frequently is the issue now of making the assumption that the experimental units are homogeneous. Because if you make the assumption that experimental units are homogeneous, look here, this is the variant, this is the experimental error versus this experimental error. So you can see that the experimental error in this particular case is too large. So we need to get a way of removing the variation due to the animals, the different type of animals. And this brings us to the second types of design, what we call randomized complete block design. So we know experimental material are always not homogeneous. If you're an animal scientist, you can have all these animals here available to you to do your experiment. Now, let's say we are interested in weight gain. We have feeds, new feeds that we want to test on the different animals. Of course, this animal, which is highly emaciated, is going to respond differently from this animal with a good body score. So unless you take this into consideration, you're going to end up with a very messed up experiment. So this is where the issue of blocking comes in. So randomized complete block design is useful, especially when the experimental materials are homogeneous. So here, there is a predictable trend in the variability of experimental material. Now, my advice here is don't design your experiment in the office. If you're going to do a field experiment, don't design it in the office. Because design an experiment in the office, you will go in the field and find completely a very, very different kind of situation going on there. Okay, so you need to go physically to the field, look at the area where the experiment is supposed to be set up, look at things like, are there shading, is there issue of slope, is there any indication that there's variability in soil fertility or the soil type, because you may find one area is rocky, another area maybe is a more deeper soil. So you need to take that into consideration. So the knowledge of expert material is very, very important as far as blocking is concerned. So you create your block, you have a block 1, block 2, block 3, block 4. So instead of assuming that these dogs are uniform, you group them up. So then later, I can then test my treatment within this group. I test my treatment within this group. I test my treatment within this group. I test my treatment within this other group. So in the field, for example, you could create like what I did, I showed you earlier on. Let me go back a bit. Yeah, so like this could be your block. So if we look here, okay, so this is your block. This is your block 1. This is your block number 2, block number 3, block number 4, block number 5, and block number 6. So you can see that from the shading, the blocks are different, but within the block, there's kind of uniformity. Now, if your block runs upwards, then you're going to have a problem, because remember, we want uniformity to be within the block. So here we then have our block 1 up to 6. So this is also going to be in the script that I gave you. I generated a randomized complete block design using this, and then it kind of you will run this like what we were yesterday doing. So here you're just creating a vector. You create this vector here, you create another vector, and then later you combine them, and then you end up with a good plan. So key things here is that treatments are randomized within each block separately. All treatments appear in each and every block, and then each block serves as a replicate. So here the number of block is equal to the number of replications. So that is what the whole thing. So the first thing is we do randomization block by block. So this is the easier way of using using ARA to randomize, to get for you a randomized complete block design field plan. So it is in the library Agricole. So we are going to, the first thing that we do, we're going to create our treatment. If we say we have 6 treatments, then we use the function. This is the function that we're using. Design.aracbd. Then you specify the treatment. You specify the number of replication or number of blocks, and then you put a seed number. Now I want to talk about the seed number here. So how do you block in a greenhouse? In a greenhouse, the key blocking issue could be the direction of the sunlight, or how sometimes if it may be hotter in the middle of the greenhouse and cooler on the edges of the greenhouse. So you can use the position within the greenhouse to actually block your experiment. So you could block for light, intensity of light, depending on where the light is coming from. If there's a potential that moisture is coming from one direction, you could also use that for your blocking. Okay, so here we have our function that design, and then we create an object which we call out.design, and then out of this object here has several things inside. One of the things that it has inside is what we call a book. So a book is literally the field book or what you actually have. So we moved from extracting this RCBD, we extracting something from the result. This result, we created this object. From this object, we are picking what we call a book, and then we do some leveling here. Okay, so what we have in the book as a block, we want to label it as block 1, block 2, block 3, block 4, block 5, and block 6. And then what we have in the treatment, remember the treatment here is 1, 2, 3, 4, 5, 6. So we want to label it as A, B, C, D, E, and F. And then finally, this is what you're going to end up with. So you can see that block 1, plot 1 will receive D, block 1, plot 2 will receive F, block 1, plot 3 will receive B, and then you have C, you have E, you have A, and then you go to the next block. The seed equals to 11 is to allow you generate the same design again and again. So if you put seed equal to 50, it will generate a different design. If you put a sheet seed equal to 1,000, it will give you another design. So literally, this is actually not a true randomization process. It picks for you from the different design that are already there, gives you one. And so the process of the picking is literally what you're having there. So you can change the seed. So if you want to show your supervisor that you generated your design using HARA, don't forget to maintain the seed, because if you change the seed, you're going to show a different thing and they will say you're lying. Okay, then the other thing is there's what we call a Latin square design. So there's what we call a Latin square design. Yeah, there's what we call a Latin. So with the randomized complete block design, we are controlling variation in one direction. So you can see from here that our intensity of the color goes down. So here, the first block has lighter color, and as you go down, the plots get darker and darker. So this could be representing for you your soil fertility status of the different areas of the plot. Now, you could also have a situation where, in addition to the change in the soil fertility, there could be some trees on the left-hand side, and those trees can provide shading, and that shading moves in a certain direction. So you may want to control both the fertility gradients. You also want to control the shading. So here then you may need to block in two different directions. So you can block for fertility gradient, then you can block for the slope. So this would be what you have. So the Latin square allows you blocking in two directions, and each direction you have actually a randomized complete block design. So here, if I look at only the slope, I have a randomized complete block design when I look at the slope. When I look at fertility gradient, I have a randomized complete block design in each case. So that's what we have as a Latin square design. So this could be, you have shading in one direction, you have fertility gradients in the other direction. So this is the same field, but we are trying to use different colors to show the direction of what we are trying to have. Now, this is Latin square can be very useful in the lab. For example, if you're dealing with machines. So let's say we have machine 1, 2, 3, and 4, and then you have also operator or technicians 1, 2, 3, and 4. So here you can see that you need to block using the operator, because each operator may have something unique with them. So you block using operator. So you can see the operator here is testing all the treatment. The operator here is testing all the treatment. The operator here is testing all the treatment. And this is also testing all the treatment. So if we ignore the machine, we have randomized complete block design. We are blocking with the operator. Now, if we ignore the operator, we can have a randomized complete block design with the machine. So here you have two independent randomized complete block design that are imposed on each other. It's also very useful in nutrition studies in animal science. So here, for example, when you're dealing with different lactation period, you have different animals. Of course, the different animals will differ in how they perform, how they biologically behave, and then the time also is different. So here you may want, you look at this animal is different from that animal, is different from that animal, is different from that animal. Now, the weeks are also different. Week 1 is different from week 2, different from week 3, different from week 4. So I have my treatments here, and I need to control using two different directions. So I have this versus that. So that's what we have as our Latin square design. I'm not going to spend time on this. So you can also generate a Latin square design using the function agricole. So here the function is this using the package Agricola. Here is the function design.lsd. That is design.latin Latin square design. The other one was design.rcbd. And then the other one, the one for CRD, you also design.rcd. So this is what looks like after you've done your design. And then I would want to skip this a bit. Let me skip this. I just want to talk about an incomplete block design briefly, and then I want to just present you some additional information on analysis of variance. Then I hand over to my colleague. So when you're dealing with randomized complete block design, the assumption is that each block can accommodate all the treatment that you are trying to study. So if we go here, okay. So here I have my block as 6 plot, 1, 2, 3, 4, 5, 6. So here means I only have 6 treatment that I can test. Now, sometimes you may have more than 6 treatment. So then a block in this case may not be complete, may not have all the treatment, will be incomplete as far as the treatments are concerned. Okay, so we talked about the importance of blocking, and we say blocking reduces experimental error and give you more precision. We always, when you are trying to do, you're trying to do analysis, you want to estimate your parameters with a very high level of precision. So that is why blocking becomes important. And then blocking also allows us to make comparison within the different conditions. Okay. There are challenges that can come from blocking. So sometimes you have a large number of treatment that require blocks that are larger, okay? But as the block also gets larger, the condition becomes more heterogeneous. And so if you try to force as many treatment as possible into a block, you're going to end up with a very big block that will no longer be uniform. Like I have this example. Let's say I'm interested in studying puppies. Then I have 9 treatments, but I only have 4 puppies. So the question is, can I borrow the neighbor's puppies? The neighbor's puppies could be the puppies of a wild dog. So now if I'm going to bring 5 of these puppies here, I'm going to have my block, which is size 9. We'll have all my treatment. But you see, I've also brought in variability in. So sometimes then it's important, it may be necessary to actually have a smaller block than having a block that takes care of all the treatments. So sometimes blocking is also based on natural grouping. So for example, you can use each mother, each piglets set as your block. Now in this case, then you can go, this one has fewer litters, fewer piglets compared to this. So that means this one can take more treatment and this one can take less treatment. So here, your block is likely to be incomplete, while here the block may be complete. There? Are you getting me? Am I clear? Can I see the link?
Yes, you're very clear.
Okay. Okay, thank you. I think the majority are getting me clearly. Okay. So in this case, then it may also be necessary to have blocks that are smaller. So then sometimes it's necessary, therefore, to put experimental unit to smaller groups. But then you then have to assemble these smaller groups in order to get a complete set of all your treatment. So when we talk about incomplete block, we can look at incomplete block based on how many blocking factors do we have. So here you can have one blocking factor, like in randomized complete block design. So you're going to have incomplete block design that are derived from randomized complete block design. But you also can have incomplete block design that are derived from Latin square. Remember, Latin square has two blocking factors, so you can have that. The other way of looking at incomplete block design is also depending on how the treatments, the precisions of the comparisons of the different treatment. So we have what we call balanced incomplete block design versus partially balanced incomplete block design. I'm going to explain this one here, and then I will move, okay? So we have randomized incomplete block design that are based on one blocking factor, and then we have incomplete Latin square that are based on two blocking factors. For balanced incomplete block design, here each pair of treatment occur equal number of times. So that means if I'm comparing A and B occurs 2 times, A and C should also occur 2 times, A and D should also occur 2 times. So each pair of treatment should occur equal number of times in the same block. While partially balanced incomplete block design, pairs of treatment don't occur together equally. So let me just show you an example. Okay, so here we have an experiment. We have an experiment in balanced incomplete block design. This is somebody was interested in testing the tomato seed germination and trying to look at the effect of temperature on seed germination. So here the treatment, they had 4 different treatments. We had seeds that were subjected to 25 degrees centigrade. We have those ones subjected to 30 degrees centigrade, those ones subjected to 35 degrees centigrade, and those ones subjected to 40 degrees centigrade. So these are our 4 different treatments. And then here, the design here is such that there is a germination chamber which is used as your experimental unit. And each run of the experiment will constitute a block. So let's say, for example, we are going to set up an experiment today. So we are going to have chambers, and each chamber will be having a certain condition, let's say 25 degrees, 30 degrees, 35 or 40 degrees. So we set this set of experiment, that is what we call our run number 1. So run number 1 is our first block. Now, for us to have a complete randomized block design, that means within each run, we should run, all the 4 treatments should be included. So in this case, we need to have all the 4 temperatures tested. But you are told that in this case, only 3 chambers are available to the scientists. You have 4 treatments, but you only have 3 chambers for doing the experiment. That means each time you run an experiment, one of the treatment will not be tested. So in that case, each run will be incomplete because you only have 3 out of the 4 blocks. So this is an illustration. So run 1 will only have 25 degrees, 25, 30, and 40 degrees. So here 35 is missing from run 1. When you go to run 2, then you have 40 is missing from here. If you go to this one here, 30 is missing. If you go to this one here, you find that 25 is missing. So the question then is this balanced? So for it to be balanced, let's try to look at this pair, 25 and 30. So 25 and 30 are here occurring once they are in this block. They are also in this block, but they are not in this block, they are not in that block. So 25 and 30 occur together in run 1 and in run 2. Now, when you come to 25 and 40 occur in run 1 and run 3. And then 30 and 40 occurs here and also occurs there. So here we can see that each pair of treatment occur together in the same block 2 times. So that gives us what we call balanced incomplete block design. With partially balanced incomplete block design, the pairs will occur different number of times together in a block. That is what the difference between the two. So you can also use ARA to generate the different types of design. So still the same Agricola package. So you can use design.bib, that is balanced incomplete block design. You fill in a few of these and then you run. But you need to clearly know what you need to, here TRT, so you need to specify the number of treatments or the treatment that you have. K, you need the size of the block. Remember here, our block only takes 3 instead of 4. ARA is the number of replication. How many replication do you want? Okay, if you don't, but in this case, you don't need to fill the number of replication because the program will have to generate in a way that you can have all the pairs coming in together. And then you can go ahead and have that. So this would be what will be generated for you. And then I already talked about partially balanced incomplete block design. So in this case, the required number of replication may be too prohibitive for you to run a balanced incomplete block design. So partially balanced incomplete block design, which requires less number of replication, may be constructed. And I already explained to you that with partially balanced incomplete block design, the number of pairs occur together in the same block different number of times. So in some cases, you may be interested in some pairs occurring together more frequently than others for reasons that are best known to you as a researcher. So this is, I'm not going to pull here. Okay, so let me just say something briefly about alpha design. So alpha lattice design is a design that is used very frequently in plant breeding. These were developed by Patterson and William in 1976. And then in the greater UK, the number of varieties and the number of replicates were fixed by a law that if you're going to do experiment, you're coming up with new varieties, we need you to have this number of replication. Okay, first run, we are going to, I want you to first run those scripts that you have, then tomorrow I can answer the questions. But by now you should already be in position to actually look for some of this information yourself. Okay. So but the requirement by then was that you need to have a fixed number of replication and a fixed number of varieties before you can go for the trial. So the large numbers of varieties would necessitate use of incomplete block design. But the challenge there would then be that there were not enough design that would accommodate the number of treatment that were available then. So these guys came up with alpha lattice design. So this one has no limit on block size. So you can specify any size of the block that you want. In most cases, you deal with blocks that are much smaller than the number of treatment that you have. Now, so the only constraint in this case is that the number of treatments should be a multiple of the block size. So the number of treatments should be equal to the number of blocks that you require and the size of the block. So that what you want is that you can finally get your smaller blocks and combine them together to create a bigger block, what you call a super block. So a super block will then be used as a super block we don't use as your replication. That would be what you have as your replication. So that is the main thing here. And we have another, we have a nice way of looking at it here. I want you to first run, and then later we can have a small discussion with that. Now, I want to run for you, just look at one particular analysis that is two-way. And then I hand over to my colleague. So there are two analyses here. I'm not going to go into one. You're going to have the one. I did an illustration for using a Latin square to actually show the different thing that will happen. I want you to first, you get the script, you get the PowerPoint, and then you go through, but you have all the scripts with you. You should be able to generate the script. So this one here, you will read first. Okay, so I want to talk about a factorial experiment. Now this one, I'm going to slow down a bit now. I was a bit running over the other one. So let me first show you some video here before I can, not a video, I'll show you some. Just hold on a bit. So I have a rather, well, it's an interesting example, just to also lighten up the afternoon so that you don't get, I know it's getting to, the week is coming to an end. Okay, so here we are going to talk about what they call the beer Google effect. So he said always when you and your friend go to the bar to drink, before you take your 6 beers, this is how the waitress looks like. After you've taken your beers, this is how the waitress looks like. So there's some sociologists who wanted to test the effect of alcohol on choices of mate in a pub or whatever it is. The idea is always that that alcohol clouds the vision, it clouds our judgment, and then you're likely to end up with a completely different story. This one I picked from somebody who's sharing it around. Okay, so I'm going to get to my presentation now. This is not the basis, this is not the presentation I'm sharing with you. I just want to have it there. So here, there's a simple example here that happens. So anthropologist is interested in the effect of beer on mate selection in a nightclub. So the rationale. So the rationale is that after alcohol consumption, subjective perception of physical attractiveness would become more inaccurate. And this is what they call the well-known Google effect, beer-Google effect. Now, the question is, does beer-Google effect depends on sex? So this is an experiment that was designed. So this person went and picked 3 females. I mean, you pick 6 females, and each of them was given beer-free, alcohol-free lager. And then another 6 got 2 bottles of beer. Another 6 got 4 bottles of beer. Then the same was done for the males. And then they would take a photograph of the person that the participant was chatting with, and then got a pool of independent judges to assess the attractiveness of the person on a scale of, say, on a scale, a certain scale. Okay, so now we start the analysis by looking at our descriptive statistics. So here, the first thing we are looking for now is we are comparing the gender, male versus female. So you can see from here that in terms of the median, there's actually no difference between male and female. If you collapse, you ignore the alcohol effect. So the male and female seems to make choices in a similar way, although there's a lot more variability with the male than the female. Now, when you ignore the gender, then you can see that no beer or 2 bottles of beer, actually there's no difference between them, but there's a drop in the attractiveness of the choice when you go to 4 bottles of beer. So we are going to our 6 bottles of beer now. With 6 bottles of beer, we might go to 0 now. Okay, and then of course the thing here is that we have 2 factors. We have gender, and then we also have alcohol, or the number of bottles of beer. Now we then need to look at the 2 of them now. So if we look at the female alone, you can see that with female, beer doesn't seem to have any effect on the choice of the male, on their rating of the attractiveness. But with the male, once you take 4 bottles of beer, I think literally everything looks like the other one that I showed you before. So you can see now, this is what we are going to look at, what we call interactions, the interactions between the 2 factors that we are looking at. So this is what we call a 2-way analysis of variance, because we have 2 factors that we are trying to look at. Of course, you need to do your summary statistics first. So you can see here that we get the data, we look at gender and summary. So here you can get the minimum for female, and then the maximum for male. So you can see the minimum score for female was 50, the maximum was 70. The minimum score for male was 20, and the maximum was 85. And remember, we saw there was a lot more variability with the male compared to the female. So you can already see it there. And you can also look at it from alcohol point of view. So this is for no alcohol. This is for 2 bottles, and this is for 3 bottles. So you can see the minimum, the maximum. So the minimum, the person who scored who got a score of 20 took 4 bottles of alcohol. Okay. So it's always good to try to put your analysis side by side. So now if we look at gender and alcohol, we look at female, none. This is how the scores were. If you look at male, none. This is where how the scores are. If you look at female, 2 bottles, this is what you have. If you look at male, 2 bottles, and then you go to 4 bottles, and so on and so forth. So it's important to do these statistics, because it will help you to see whether there's any error in the system, or or whether whatever you have makes sense. Let's say, for example, we get a negative score. And remember, we are supposed to be from 0 to 100. So if you get a negative score, that means there must be something not right with the observation that was all the data that is given to you. So you may need to cross-check that. So when you look at the male with 4 bottles of beer, you can see how small the values are. Okay. So now we have 2 factors here. We have gender and then we have alcohol. So we can do our one-way analysis of variance. So let's say, for example, here we have one-way analysis of variance. So this is gender. The one on the right is, on the left is gender. So here is the p-value, 0.35. So on the chart, was there a significant difference between the males and female? Okay, you can say there is no significant difference between males and female. A few people still say yes, but at least the p-value is greater than alpha. So there is no difference between male and female. Now let's look at the alcohol. So the p-value, is there a significant difference between the number of bottles taken? Can I see in the chart? Yes. Okay. So here the p-value is less than 0.05. So there's significant difference. Now, this does not tell you which bottles are different from the other. That's where you need to go further down and look at the post hoc. So in terms of, you can plot from the model, from each of the model that we have generated, we can plot what we call the effect. So you can see from here that the male, female versus the male, the slight difference between the male and the female. But you can see from here that their confidence interval overlap. So that's why in this case, there's no difference between the 2 genders. Now, when you look at alcohol, you can see that there's no difference between none and 2. Actually, there's even a slight improvement with 2 bottles of beer. There seems to be better choices than with 1. But you can see the drop in 4 bottles. So these are the things. These are the things that you need actually to look into your data to be able to actually tell which direction you're going. So we first started by running single analysis, one-way analysis for this and for that. But remember, the reason as to why you are doing a 2-way, the reason why you're looking at 2 factors is because you're trying to see whether there's an interaction between the 2 factors. So the best analysis would be to look at a 2-way analysis of variance. So with the 2-way analysis of variance here, you need to have the gender and alcohol both in the model. So you can see from here that gender was not significant. The p-value for gender is 0.16. Alcohol is significant. And the interaction between alcohol and gender is also significant. Now, once the interaction is significant, that means I cannot interpret the results for the 2 factors independently. I need to combine to interpret them together. So here is then what you do. You look at the female separately, compare your bias for the female, compare yours for the male. So this is where the 2-way interaction comes in. So you can see with the female, there is actually no difference. But with the male, there is a big difference between the none and 2 versus the 4. So this is what you have as your 2-way analysis if I compare to the 1-way. You see, we have 1-way. This one only gender, this one only alcohol level, but this one combined both of them now. So we have for female, this is the situation. For male, this is the situation. So here we are saying that for female, well, you can go to a bar and you will always still come up with a good-looking man. irrespective of how many bottles you've taken. But with a man, you have to be careful. After 2 bottles, you better come back another day to make your choice. Okay, so that is how the two-way, how the interaction is interpreted. The interactions mean if I'm looking at alcohol, I need to look at alcohol with respect to the gender. If you're female, you're safe. If you're male, after 2 bottles, come back the next day, and then you can have your, yeah. So this is the same you do with varieties, with fertilizer, whatever you're trying to study. You want to be able to, you're always asking yourself, is there interaction between the 2 factors? If the interaction is there, what does it mean? If you're going to advise farmers, what do you need to tell them? Are you going to tell farmers that, please, if you have fertilizer, please, this is your variety. If you don't have fertilizer, please, you need to plant your local variety that you've been doing before. Don't waste your time with a new variety. Okay. So briefly, I introduced the concept of contrast. Now, contrast is a very, very interesting and a very useful comparison that you can make. Now, this is mainly useful when you're comparing treatment with some kind of structure. So instead of like what we saw yesterday in the analysis with the LSD, where you had the numbers A, B, B, and C. So here you are not comparing one treatment versus another. You may be comparing a group of treatment. You're comparing groups of treatment. So for example, one contrast that you may be interested in, you want to compare alcohol, no alcohol versus some alcohol. Okay? So no alcohol versus some alcohol. Contrast 2, you may want to compare 2 bottles versus 4 bottles. And then contrast 3, you may want to compare male versus female. Now, the male versus female, you can, I mean, is the same as what you have before. But you see the first 2 contrasts are slightly different. So you can look at alcohol, no alcohol versus some alcohol. Remember, some alcohol is an average of the 2 bottles and the 4 bottles. Okay, so here then, the challenge here is you need to be able to tell ARA the type of contrast, to call the contrast that you want to test. This is where the challenge is. Now if we look at, let's look at our, so here we have none, we have 2 bottles, we have 4 bottles. So now see here, none has -2, and then 2 and 4 have 1, 1. So remember, we are comparing this versus the 2. So that's why now this is how you generate the contrast. So I will multiply what I have as none with the 2, and then I compare with the other 2. So this one will give us a contrast for none versus some alcohol. Remember, some alcohol is the first contrast here. No alcohol versus some. So here I have no alcohol, I give it -2, and then +1 for the other one. The idea here, the main thing here is that the sum of the coefficients should be equal to 0. Now, after I've done with the none, when I go to the second contrast, the second contrast said 2 beers versus 4 bottles. So in that case, I'm no longer interested in the none. I give it a 0. Then I compare the 2 versus the 4. I give -1 and +1. Then what I then need to do is then I can do the analysis now. So this is my analysis, the one on this side. This is the one that I've done. I have my gender, I have my alcohol, I have my interaction. Now, this is the one down that I have my contrast in. So the first thing is it tells you gender 1. So gender 1 mean I'm comparing male versus female. And you can see from here, male versus female, the p-value is 0.1661, which is greater than our alpha. So in this case, we can conclude that there's no difference between male and female. Okay, now let's go to alcohol 1. Alcohol 1 is the one that is comparing none versus some. And then, so none versus some, you can see is significant. Then alcohol 2 will compare 2 bottles versus 4 bottles. And this is also significant. Now, the most interesting one here is you're comparing gender 1 interacting with alcohol 1. Now, gender is male versus female. Alcohol 1 is none versus some. So here we can see there is a significant effect. So what does that mean? Okay, that means if I'm comparing no alcohol versus some alcohol, I need to take into consideration the gender. Because in that case, if you compare no alcohol versus some alcohol, for female, there will be no significant difference, but for male, there will be significant difference. And the same is for alcohol too. So if I'm comparing male versus female, I will find out that 2 bottles versus 4 bottles for female is not significant, but for male, it will be significant. And then, so this one brings us to what we call the simple effects. So the interactions between 2 factors implies that the effect of the 2 factors cannot be interpreted independent of each other. So you cannot interpret the number of bottles independent of the gender. So in this case, you can do what we call a simple effect. So a simple effect is you're comparing the level of one factor when holding the level of the others constant. So more or less, this is what we are talking about. You're comparing the level of alcohol while holding gender constant. So if we look only for female, this is the comparison. If we look only for male, this is what we get. So that's what we call the simple effect. Okay, so you compare male and female at 0 alcohol level. You compare male and female at 2 bottle level. And then, so you can also construct this simple effect as a contrast. It's a bit, it needs a bit of getting used to, but the concept is very, very simple, like what we have before. Unfortunately, there's no way around this. You need to be able to construct this yourself. Okay, so here we have, first of all, we have our female, no alcohol, female, 2 bottles, female, 4 bottles, male, none, male, 2, and these are the categories that we have. Now, so alcohol effect 1. Alcohol effect 1, we want to compare none versus some. Now, none, there are 2 nuns here. There's none for female, and then there's none for male. So you can see these 2 will constitute 4, and then the other one will constitute 1, 1. So here you still have 0 as your, the sum. Now, alcohol 2 effect. Here we have 2 bottles versus 4 bottles, but there's 2 bottles for female and 2 bottles for male. They carry the same sign, and then the 4 bottles for male and female, they carry the, so you combine these 2. And then here you want to look at gender, that is gender no and gender none. So you look at female with none and male none. So this one just deals with that. And then you look at gender 2 bottles. So you compare male and female at 2 bottles and gender 4 bottles. So the hardest thing with the contrast is how do you construct this? Once you know how to construct this, then the rest will be a walk in the park. So this is the result. I think we're almost coming to the end. Okay. So you can see this is now completely a different kind of analysis like what some of you are used to. So here we can see alcohol effect 1. So alcohol effect 1 compares no alcohol versus some alcohol when you lump up the male and the female together. Effect 2 will be some alcohol, 2 bottles versus 4 bottles. And then gender none. Now see the gender none. Gender none is not significant. What gender none means is that if you compare male and female, when they have not taken any alcohol, when they've taken 0 bottles, their choices are not significantly different. Now, even with 2 bottles, the choices are also not significant. But with the 4 bottles, the choices become significant. So this is what you can now do with your data set. You can break up your data, your analysis, and somebody can look at you and say, well, this person knows what he or she is doing, and you can bias the examiner in one way or the other. But if you write a thesis like everybody else, then you'll have a problem. So you can still go ahead and run the post hoc like what we did before, but I think I want to stop here so that I give my colleagues an opportunity to start. Maybe we can have 4 minutes break, and then we can have 4 minutes break, 10. We have 10 minutes break, and then we come back and at 3:36. So 3:47. Come back at 3:47. Okay, thank you.
start. Okay, they've already switched off. Okay. Mhm. Okay. Good afternoon, ladies and gentlemen. I believe you're back from the break, and we are going today to talk about correlation and regression with R. Are we together? Can I see in the chat? Are people back? Are we back? Should we start? Okay. Okay. That is great. Thank you very much. So as I've been saying, we are going to talk about correlation and regression. And we'll start with the theory. Then once we finish theory, we'll do practicals. Correlation is a measure of degree of linear association between two variables, and it ranges between -1 to +1. We find that sometimes you have what we call positive linear correlation, and the general trend is that points run from bottom left to top right, or negative correlation, where the points run from top left to bottom right. Then if you have just a random scatter of points, then we call that no correlation. The correlation coefficient gives us the type and strength of linear correlation. It is denoted by a small letter r. As I've said, it ranges from -1 to +1. So if it is -1, it's called negative correlation. If it is +1, it is called positive correlation. Okay. So if it is close to 1, if r, small r is equal to 1, then that is called perfect positive linear correlation. And if it is equal to -1, it is called perfect negative correlation. If r is equal to 0, then we call that no linear correlation. With the correlation coefficient, we are able to quantify the strength, that is the magnitude of the correlation, and direction. Direction is positive or negative. And if we are dealing with continuous data, then that type of correlation is called Pearson linear correlation. And we have a formula there, which you can refer to later. Sx means sum of squares for x values, and Sy means sum of squares for the y values. Graphically, we find that if there is a strong correlation, the points tend to lie along the line that goes through the 0, point. So if you look at the topmost graph, we see that most of the points are concentrated along that line. And if we draw a line through this point, we'll see that most of the points will fall on that line. That shows that there is a strong relationship between weight and chest girth of the birds. Now you can see that at the bottom left corner, it's just a random scatter of points, and the correlation value is 0.07, indicates the correlation is quite weak. Then we have the bottom right corner. The points are scattered, but not as in the previous graph. And we see that it's not very strong correlation, and the magnitude of the correlation is 0.67. You can see all these ones are positive correlation. That's why the points are running from the bottom left to the top right. For negative correlation, we will see that the points run from the top left corner to the bottom right corner. That is negative correlation. And we see that if the correlation is strong, the points are close together. Compared to the graph where we have a random scatter of points. And this other graph where R is minus 0.62, we find that the points are spread apart, but not as in the graph where the correlation is minus 0.13. So with the graphs, the scatter diagram can always tell you what magnitude that type of relationship has. Correlation is not causation. What do we mean? Just because two variables are correlated does not mean one causes the other to change. So, does religion cause crime? In most cases, you find that if we get data on people who have been found with bombs on their body, they are said, I'm not very sure, we need to confirm, they are said to be Muslim. But does it necessarily mean that being a Muslim causes you to tie yourself with a bomb? No, it doesn't. Or if we look at the marble stocks, we have a hospital here called Mlago Hospital, and there is a tree full of marble stocks, and they are giving birth every day. But even the women are giving birth every day. Does it mean that the marble stock causes the women to give birth or the other way around? No. That's why we are saying that correlation is not causation. So when two variables are highly correlated, it does not necessarily mean one causes the other to change. So for us to see causation, we need to perform an experiment. So Pearson correlation coefficient measures the strength and direction of a linear relationship, but not the causal relationship. In R, we can compute correlation using the command COR, C-O-R, in bracket, you put in the two variables. So with that command, we are able to compute the correlation coefficient, and then COR.test, it tests the association between the paired samples. So you get the correlation value and the p-value. So you're able to know whether the correlation coefficient that you've obtained is significant or not significant. So when you write commands in R, you also specify the method of correlation that you want to use. So if X and Y are continuous, then we specify the method as being Pearson. And if we have rank, then we specify as being Spearman correlation. It is say X and Y are numeric vectors with the same length. So in most cases, when you have a data set where X and Y are different lengths, then you're going to have an error in your console. So we've already talked about the Gigi scatter. It is used to obtain a scatter plot. But on top of the Gigi scatter, we can obtain the correlation, we can add a regression line, we can specify the method of correlation, and also specify the title for the X axis and the Y axis. So in one go, we can have a correlation coefficient computed, a regression line, we can attach confidence interval, the correlation coefficient, we specify the method, and also label our X and Y axis. So on the right, we see the graph that comes out after you've told R to use the Gigi scatter. So on top here, we see the R values, the correlation coefficient, and we also see the p-value, which tells us, because you can see it is 2.2 to the power minus 16, it is smaller than the significance level normally used for agriculture, environmental studies, etc. And we conclude that the two variables, price and weight, are strongly positively correlated. So we see, if we look at this result here, maybe it's not, it should be visible in your notes. If you look at the value of R is 0.92 and the p-value is 2.2 times 10 to the power minus 16. So the null hypothesis here is that no correlation between price and weight of diamond, and the alternative is that there is a correlation relationship, or the two, price and weight, are strongly associated. In R, again, you can perform or conduct a correlation coefficient by specifying an object name, which is ASSS. Then you assign to core.test, which is the command for computing correlation between diamond and price. And the method you're using is the Pearson correlation. So the results that you see after printing the object is down here. It gives you a t-value with 98 degrees of freedom and the p-value, which you've already seen. It also tells you that the alternative hypothesis is true, meaning correlation is not equal to 0. When correlation is equal to 0, it means the two variables are not associated with each other. So on top of that, it gives you the confidence interval for the correlation value and also gives you the correlation coefficient. Okay. So the next slide is talking about interpreting the correlation value in details. So we see that our p-value of 2.2 times 10 to the power minus 16 is less than 0.05. That is a significance level. And we conclude that weight and price are significantly correlated with a correlation coefficient of 0.92. And it is positive. Okay. We could sometimes produce a correlation matrix, and it used to investigate dependence between multiple variables at the same time. So if you have more than two variables, we can construct a correlation matrix and be able to obtain the correlation between different variables. Now, the package we need for this type of analysis is called HIMSC, HMISC package, and it produces the p-value and the correlation coefficient. So we'll see later when we are going to do practicals that in case you don't have that package, we can install it. So let me see in the chat. Are we fine? Should we continue? Do you have any problem? Sorry, my internet has a problem. Sorry about that. Okay. 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 Thank you very much. Just mind the speed, okay? I'm going to switch on my speed governor so that I can reduce the speed. Okay, okay then. Thank you. And in the Q&A, please kindly send material. My colleagues will send the material, the link for the material to you. Okay, so let's proceed. So if a pair of variables have significant linear correlation, then the relationship between the data values can roughly be approximated by a linear equation. The process of fitting the linear equation to the data is known as linear regression. Yeah. Okay. Okay. Okay, so the material where we find the PowerPoint is under day 3. So when you go in day 3, week 2, day 3, that's what Helen is telling me. That's where she placed that information. So go to the same link, but look for week 2, day 3. Okay, so the process of finding a linear equation that fits our data is known as linear regression. And the line of best fit is called the regression line. Now, regression is concerned with a relationship, the study of a relationship between variables with an objective of identifying that relationship. When we talk of identifying the relationship, we want to know, is it a linear relationship? Is it nonlinear? Is it curvilinear? Estimating. When we talk about estimating, we are looking at the parameters, the slope and intercept, and then validating the relationship. Remember that the other objective of regression is prediction. So we need to validate the relationship to see whether the assumptions under the regression analysis are valid for us to be able to use the regression model that would have established. So that is the use of regression. The linear relationship is a straight line model consisting of the intercept B0 and the slope B1. The slope of the line indicates that for each unit increment in the x, which is the independent variable, then y increases by the slope, the value of the slope B1. And B0 is the intercept. When x is equal to 0, y changes, or y is equal to the value of the intercept. So when x is equal to 0, y is equal to the value of the intercept. And when x increases by 1 unit, then y increases by the slope B1. If the slope has a negative value, then we are going to say that when x increases by 1 unit, y decreases by B1. So that's how we interpret our straight line model. So this is what I've just been saying, that the slope represented by B1 and describes the change in y for every unit change in x. And y is the value of B0 is the value of y when x is equal to 0. So the line that we fit onto our data is called the line of best fit. And once it is fitted to the data, not all the observed values fall on that line of best fit. Now, the difference between the observed value and the predicted value, which we are going to talk about in a minute, is called residue. So my colleague has been talking about residue, but talking in terms of experimental design. When we conduct an experiment, we find that we have 100% in the response. When you fit the treatment onto the model, it explains some variation, and whatever remains is what we call residual. The same applies to regression. When we run a regression between y, the response, and x, the independent variable, it explains some variation in the response, and whatever remains is called residual. Now, the line of best fit minimizes the sum of squared error terms, and it is obtained by the method of least squares, which we abbreviate as LOS. Now, with the method of least squares, the slope and intercept obtained by LOS, those two are called the least square estimates. So we have a regression line here, and that is the blue line, which has y hat, and the red points are the observed values, which are called y, the response. So when you fit the line, then the model becomes y hat is equal to beta 1 x plus the intercept beta naught. But you can see that this line, although it has fitted most of the values, the red points, not all of them fall on the line. So the difference between the red points and the blue line is what we call residue. So let's go and see graphically what the residue is. Okay, so when we look at this graph, the bigger graph here is what we had before, and you can see the line going through the red points, but not all of them fall on the blue line. Now, if we magnify this, we see that the blue line is the model line, which equation is y hat is equal to beta 1 x plus beta naught. The red points here have an equation, y is equal to beta 1 plus beta naught plus epsilon unexplained variation. So the difference between the red points and the model line is the residue, which is epsilon. So you can see here in the diagram that we are pointing onto the blue line and the red point. So the difference between that gives us the residue. The red points are the observed value which you collect from the field, and the predicted value is y hat. So the difference between y and y hat gives us the residue. The ordinary least squares regression, which is we are abbreviating as LOS, minimizes the sum of the square differences between the observed and the predicted value. Now, the best fitting line, which is our blue line, has the small sum of squared residues. Now, ordinary squares or LOS estimators can be estimated using the formulas given, and those formulas are obtained through differentiation, which we will not do here. So when we have the original model of the observed value is equal to beta naught plus beta 1x plus epsilon, we now get the difference between the fitted line and the observed, and then square those residues, then differentiate with respect to beta naught and beta 1. That's how we come up with these formulas. So for now, we will know these formulas. We don't need to know how to differentiate, because then we will use R to give us the value of the slope and intercept. But I know that those who are doing pure statistics do differentiation and come up with these formulas. So that's the intercept is equal to the mean of the y values minus the slope into the mean of the x values. Meanwhile, the slope is equal to the sum of the cross product between x and y over the sum of squares of x. That gives us the slope. So down here, we've expanded SXY and SXX. And we also have the formulas for the mean for x values and the mean of the response. Alternatively, option B, we can obtain our slope in this expanded format, but you end up with the same values. So here we are still talking about our residues. We are saying that residues are very useful. They allow us to calculate the error sums of squares. And when you have the error sums of squares, we use them to compute what we call coefficient of determination, R squared. And we'll see that R squared tells us how much variation has been explained in the response as a result of the regression between the response and the independent variable. So R squared, as I've just said, tells us the proportion of variation in the dependent or response variable that has been explained by the model. Below here, we have formulas for computing R squared. And R squared is equal to 1 minus R. This is the regression sums of squares over the total sums of squares. Gives us R squared. And we have also a formula for regression sums of squares, which we are not going through now. So R squared is very important when you're doing model building. If a model explains above 70 of the data variability, then that model fits our data. We normally say the model explains a big proportion of the variability in the data set. When R squared is near 1, then the model fits the data well. And when R is near 0, then the model poorly fits the data. We'll find that when we do our analysis, we'll have R squared and then adjust R squared. Now, R squared is what we've seen. It is the ratio of explained variation over total variation, or it is the ratio of the regression sums of squares over the total sums of squares. That gives us our R squared. Then adjust R squared. It is the value of the variability explained, but then it adjusts for every parameter added into the model. The R squared is adjusted automatically. So if you have more than one independent variable in the model, the R squared will adjust the number of parameters in the model. So we see that if we have only one response and one independent variable, we are going to have only 2 parameters. But if we add another independent variable, we will end up with 3 parameters. So the R squared will be adjusted. Meanwhile, R squared alone is inflated as we add in more variables in the model. So when we are doing model building, we quote R squared adjust because it's going to adjust for the number of parameters added in the model. It is not affected by you adding more variables in the model. Meanwhile, R squared alone keeps on increasing and increasing as you add variables in the model. So when you're doing model building, take care of that to make sure that you quote R squared adjust. So what are we interested in when we are doing regression? The other thing I forgot to mention that if we have one response and one independent variable, that type of relationship is called simple linear regression because we have one response and one independent variable. But later we'll see that when we have one response and many independent variables, that is called multiple regression. And then if we have many response and many independent variables, then that type of analysis is called multivariate regression. Okay, so for now, we will look at simple linear regression and multiple linear regression. And if time allows, we are also going to do model building. So when we are doing regression analysis, we are interested in the slope. When the slope is equal to 0, then there is no linear relationship. Okay, let me see whether I can get our pen here and we see. Okay, so when the slope is equal to 0, this is what happens. The relationship is like that. It's just a straight line, horizontal straight line. This is when beta 1 is equal to 0. Now, if the relationship is not equal to 0, we can have a positive relationship or we can have a negative relationship. It depends on the type of relationship. So we are interested in testing the slope and finding out whether it is different from 0. So our null hypothesis here under regression is that there is no linear relationship between the response and the independent variable. And alternative is that there is a linear relationship between the response and independent variable. We can test that hypothesis using a t-test and a regression, and we are testing the slope, and our target value is 0 over the standard error of the slope. So this is the standard error of the slope. And the degrees of freedom correspond to the residual degrees of freedom, which is n-2. Or we can use an f-test, which is a ratio of the mean square regression over the mean square error. We are going to see that later in the ANOVA table. Where do we get mean square regression, mean square error? Okay. So far, do you have any problem, or I continue in the chat? Can I see, do you have any problem? Is there anything that needs clarification in the chat? Okay. Twende sana. Okay. Degree of freedom. Okay, let me talk about the degree of freedom. Okay, great. Okay, now, when we perform an analysis on our data, let me pick the, so when we perform an analysis, we will have what we call ANOVA table. You've heard of ANOVA table and experimental design. There we have a categorical variable for the independent variable. Here, our categorical variable, our variable is continuous. So we'll have what we call degrees of freedom. Degrees of freedom. Degrees of freedom, that is the amount of information that is free from constraint. So source of variation, I'm going to represent is that as R regression. Then we have residual or error, I'll represent it as error. And then we have total. So let me put a line to separate these two. So when we have our linear model, the response is equal to the intercept beta naught. And then we have also the slope beta 1 x. In this model, we have only two parameters, the intercept and the slope. So the regression degrees of freedom is equal to the number of parameters, which are 2 minus 1. Then when we go to error, the degrees of freedom is equal to the total number of observations minus the number of parameters. That is number of P parameters. When we come to total, is equal to the total number of observations minus 1. Okay, so that is degrees of freedom. Here, when we come to the total, if you look at the formula for total sums of squares, we require the mean before we can actually compute the total sums of squares. So that's why we have a penalty of 1. Here, error sums of squares, in order to compute error sums of squares, we need to know our intercept and slope. So you are using 2 degrees of freedom. I mean, you're using 2 information to compute the error sums of squares. That's why our degrees of freedom is n minus the constraint, which is the number of parameters, 2. And here it is the number of parameters minus 1. Or the regression degrees of freedom also is equal to the number of independent variables in the model. Here we have only 1 independent variable, and therefore the degrees of freedom is also going to be equal to 1. So that is roughly what degrees of freedom is. Okay. Okay, so let's go on to the next slide. Okay. So when you analyze your data, this is the output that you get. So the source of variation here is regression, and we have residual and total variation. We've already said that because we have only one independent variable, our degrees of freedom for regression is equal to 1, or the number of parameters which are 2 minus 1. Here, we know that our total observations will be 30 plus 1, because we know that the degrees of freedom is n minus 1. So we'll get 31 observations. So the residual is going to be n minus 2, which is 31 minus 2 gives us 29. Sums of squares, there's a formula, but we won't go through those details. I want us to talk about the mean square. Now, for regression, the mean square here is the ratio of sums of squares and degrees of freedom. So when you get the sums of squares, divide by degrees of freedom, we get the mean square here. The same applies to residual. Residual mean square is equal to the sums of squares for residual divided by the degrees of freedom. We get the mean square of 18.1. The other thing we are interested in is the F value. The F value, which is the F test, is the ratio of mean square regression and residual mean square. So this value here, we get 7,581.8 divided by 18.1. You get this value. And our p-value, we can use the principles of mathematics to calculate it. So that is the analysis of variance table under regression. Now below here, we also have some output. We have S. S is the square root of 18.1. So you can find out whether that is true. You can get your calculator, get the square root of 18.1, gives you S, which is the standard deviation of the data. We talked about R squared, and I said R squared is the proportion of variation explained in the response. In our analysis, our response is volume, and our independent variable is diameter. So 93.5% of variation has been explained in volume by the regression model. R squared adjusts, you can see that it has already been adjusted. It adjusts for the number of parameters in the model. We have two parameters, the intercept and the slope. So it has the same interpretation that 93.3% of variation in volume has been explained by the regression model. Okay, so that is the interpretation of this output. This one you should know. The p-value. Remember we said we are testing the slope. The null is that there is no linear relationship. The alternative is that there is a linear relationship. So we can use the p-value or the f-value. So the p-value here is smaller than 0.05, which is our probability that the null is likely to be or assumed to be correct. So if you get any value smaller than 0.05, you have evidence to reject the null and conclude that there is a significant linear relationship between volume and diameter. So that is the output here. Now let's go to the model. So this is our regression model, and we have a negative intercept. This negative intercept, sometimes it makes sense, sometimes it doesn't make sense. For example, here we are talking of trees and their volume. So if we say that when there's no diameter, the tree has a negative volume, it doesn't make sense. So when we are interpreting the intercept, we need to be a little bit careful. We have to look at the biological system before you interpret the intercept. However, the slope here indicates that for every unit increment in diameter, volume increases by 5.07 cubic meters. For every unit increment in diameter, volume increases by 5.07 cubic meters because the SI unit for volume is cubic meters. Any questions so far in the chat? Please, Prof, between R squared and adjust. Okay, which one should we report? Between R squared and adjust. If we are doing a simple linear regression, you can report any of that too. Okay, just like you can either report 93.5% or 93.3%. But if you're doing multiple regression, we report the R squared adjust because it is not influenced by addition of variables in the model or addition of any additional parameters in the model. Okay. Any other question? Would you repeat? I'm really enjoying this. Okay, thank you. I don't know what to repeat, but let me repeat what I've just said last. I have said that if you're conducting a simple linear regression, which is a relationship between one response and one independent variable, you can use either R squared or R squared adjust. But if you're doing multiple regression, you're better off using R squared adjust because it adjusts for any parameters added in the model. Okay, even when we go to do model building, you will see that we'll be looking at R squared adjust because it's not influenced by adding more parameters in the model. Okay. Okay, so we continue. So this is what I've been talking about, that ANOVA test is testing this hypothesis. And if we are using the F test, we should have what we call F tables. If you go online and put in F distribution tables, they consist of the numerator and denominator, degrees of freedom, and then a value that intersects those two gives you the what we call a rejection region value or critical value. So here I've indicated the critical value as 4.18. If you compare it with the calculated value of 419.36, the calculated F value is greater than the tabulated, indicating that there's evidence to reject the null. If we use the p-value, we found that our p-value of 0.00 was smaller than 0.05, indicating strong or sufficient statistical evidence that there is a linear relationship between volume and diameter. Okay. So within the notes, there are examples. So in your free time, you can go and actually attempt this exercise. It is about phosphorus, inorganic phosphorus, and then phosphorus content in the corn at the end of the season. So we have assumptions. Anytime you run a model, any model, there are assumptions that we make. And under simple linear regression, the first assumption is that Y is a random variable normally distributed with mean mu and variance sigma squared. So why do we say random variable? We assume that you collected your data randomly. It's not subjective. And the data you've collected follows a normal distribution. Then the other assumption we make is that the unknown random errors, the unexplained errors or residual we've been talking about, are assumed to be independent, normally distributed with mean zero and constant variance sigma squared. I've misspelled squared here. You can correct it in your notes. And then we also assume that the random errors have constant variance. We also assume that there is a linear relationship between the response and the independent variable. So those assumptions should not be violated when you're running your regression, so that when you come to prediction, you're able to do predictions with a valid regression model. How do we test those assumptions? We use the residuals. Remember the residuals, and we call them unexplained variation. We call them error. Those are the ones we use to test the assumption. Normality of the response, we construct a histogram of the response. That if it comes out to be bell-shaped, then the response has normal distribution. Now, for linear relationship between the response and the independent variable, we construct a scatter diagram. If you find there's a linear relationship, then truly the relationship between the response and the independent variable should be linear. When it comes to the residuals having normal distribution, we construct a histogram of residuals, as we are going to see. So statistical inference can be made when the postulated model is adequate. And a correct model should have residuals assumed to be normally distributed with mean 0 and constant variance. And they should not also be correlated. So independence of residuals means that the residuals are not correlated. When residuals are correlated, it implies that there is some dependence in your data, which needs to be explained either by another variable, time, or you need to use another model to explain the correlations. So a histogram of residuals is used to check for normality. So we all know about normal probability plot. They are also used to test for normality. So a plot of residuals versus normal score helps us to test for normality, as you shall see in R. And then for us to check for constant error variance, a plot of residuals versus predicted values. So if there's a random scatter of points, as you see here, that indicates that the residuals have constant variance. Anything that deviates from the random scatter of points, then we have to pay attention to the pattern that we see in the graph. So what you see here is just a random scatter of points. And I've said it shows that the residuals have constant variance. Now, if points from a horizontal band are around 0, if points form a horizontal band and they're just randomly scattered around 0, then the assumption of constant variance is valid. If the residuals increase with increasing fitted value or response, then let's draw the graph. If we see a pattern like this, I hope you're seeing this graph here. So the residuals have a mean of 0. Here we have epsilon, which are residuals. And here we have fitted value y hat. So if we get something like this, I'm not very good at drawing art, but we are seeing that these things are increasing with increasing or decreasing with increasing fitted value. It is a ball-shaped, a funnel-shaped curve. You see a funnel-shaped curve? That shows that the assumption of constant variance is invalid. So that is the first pattern. If you observe that, you know that that assumption is valid, and you may need to transform your data to normalize the variance. So normally, we use the natural logarithm of the response. And once you apply natural logarithm to the response, you're able to deal with this type of pattern. Then let me show you another pattern. So that is when we have a funnel-shaped curve. How about when we have something like this? Okay, let me draw here. It is okay. So if we have something like this, a ball-shaped, so I'm going to use dots. Hope they are visible. You see this ball-shaped? Is it visible? So you can see this is a ball-shaped. This is our epsilon, and this is the fitted value. Or sometimes you can use the response itself. That shows that your response has a binary nature. It has the positive side and also has the negative side. And because of this ball-shaped, the linear model doesn't fit your data. So you need to apply a logistic regression instead of simple linear regression. Okay, so that is another way to know that your data does not have constant variance if you see a ball-shaped. The other, let me erase this. The other formula, a graph that you can see is this one. So we have our y hat, the fitted values, versus the epsilon, and this is the zero line. If you see a curvature, so we are going to see, if you see a curved pattern like this, this shows you that actually your linear model doesn't fit the data. The assumption of constant variance is invalid, and you need a curvilinear relationship to explain this relationship between the response and the independent variable. So those are the things we should look out for. Look out whether your model, your linear model fits the data. If you see this, then your linear model doesn't fit the data. You need to run a curvilinear relationship where you have powers on the independent variables. If you have a ball-shaped, then you need to run a logistic regression. And if you have a funnel-shaped, then you need to log transform your response and then rerun the regression and check the assumptions. So the last assumption is independence. Now, if you have time, if you've collected data over time, that data normally is correlated over time. So you will find that this assumption of independence is violated. So how do we see that? You will see that when you construct your graph, you're going to have something like this. You're going to have points running like this. Okay, so this is going to be your pattern of points. This is our epsilon, and this is y. And this shows you that there is time factor in your data, which you need to take into account. And the variance is increasing and decreasing as well. So the assumption of constant variance is violated, and also the assumption of independence is also violated. So in that case, then we are going to run, either we are going to perform time series analysis, or you are going to include time as a factor in your model to account for this type of relationship. Okay. Any question? Any query? Let me see in the chat. Should we continue? What is a curvilinear relationship? Okay. Okay, when you have, let me get the pen. If we have y, sorry. Okay, so here we are. And apparently I can draw. Okay, so now if you have this pattern here, and this is our y, and this is x, it means that our y has a relationship between beta naught plus beta 1 x, but again, to account for this curvature here, we have to apply the x squared term, the quadratic term, or you could even apply the cubic term. So we could have another, depending on what is significant. So that is a curvilinear relationship. Nonlinear relationship, remember, in that case, we'll have something like this. We can have either going like this, or coming down, but in the same format. So when we have a nonlinear relationship, it means that the relationship between the response and the independent variable is linked in a nonlinear format in terms of the parameters. You see here we have a straight line model. In that case, our model is going to be something like exponential beta naught plus beta 1. So we will no longer have a linear relationship, but we are going to have a nonlinear relationship where the parameters are linked onto the response through a nonlinear format. Okay. So we have curvilinear where we have powers, and in the regression jargon, they call it a polynomial model, and we have a nonlinear model. Those are the difference between curvilinear and a linear model. Okay. In the linear model, we only end at beta naught plus beta 1 without powers. When we have a curvilinear relationship, the independent variable is accompanied by powers, either a quadratic or a cubic or x to the power 4, depending on what is suitable for your data. Okay, let me see in the chat in case there's any question. And when is a multinomial regression done? Okay, mult means many. Nomio means nominal, which is a categorical variable. And we can do that if your response and independent variables have categorical nature. So if your response is just names, for example, agree, disagree, etc., then, and you want to regress agree, disagree onto age, sex, etc., then you can run a multinomial model. Why logit model, not probit? Now remember, logit regression here, we are looking at a relationship between our continuous variable and independent variable. A logit probit model is used when you have percentages, you have proportions, not when you have continuous data. If the residuals show that there is no relationship between the variable, how can we get, oh, okay. The relationship already can be shown, as I've indicated. In the residual, we can have a curvilinear relationship showing the residual. Automatically, you know that you need to run a polynomial regression. And in case you see a bowl-shaped, then automatically you know you have to run a logistic regression. If you see you have a funnel-shaped, then you know that you just need to apply natural logarithm to your data, and then that bowl-shaped is corrected. Okay, any other question? Those are the ones that I found on the way. I don't know whether there is, okay. Okay, let's go ahead. What's the time? Okay, we have 13 minutes to go. Let me just erase and see what we can do in the next 6 minutes so that I leave a few minutes for closing the session. So when we go to multiple regression, you will see that the assumptions under simple linear regression also apply to multiple regression. And in addition to that, we'll see that there's an extra assumption. So we normally do the same tests. Okay. So here, I've already talked about this natural logarithm, or you use the square of either the response or independent variable if you find that the assumption of constant variance has been violated. And I talked about polynomial regression, which is a special case of regression where powers of the independent variable play a role of individual variables. So I have a data set. Among the data set you have, there's a data set that has a polynomial nature, and we will look at that data set and see when you analyze ignoring the curvilinear, how the relationship looks like. And after you've recognized that there is a curvilinear and include the quadratic term, what happens to your assumptions, okay? In the few minutes left, let's talk about multiple regression. Now, we've said that multiple regression is a relationship between one response and many independent variables. Sometimes you see that we've run a simple linear regression, but you still have factors in your data set, and the unexplained variation is so large, meaning that your R squared is so small, maybe even 30% of your model. So if you find R squared is so small, then that means that there are other factors that you can add into the model to explain the variation in the response. So if you have a model where you have one response and more than two predictors, that is called multiple regression. And the multiple regression model, you can see we have an intercept slope for each independent variable. And the slopes are called unknown parameters and intercepts. Epsilon is the random error, or we call it residual or unexplained variation due to other factors not included in the model. Then assumptions mentioned under linear relationships still hold. And the last assumption is that the independent variables should not be correlated. If they are correlated, the correlation should be weak. So X1, X2 should have very weak correlation for you to have a valid multiple regression. In case you find X1, X2 are correlated, then we can run a multivariate regression. Or there is a method called centering, whereby you get the average of X1 and subtract from X1, that's what they call centering, and then rerun the regression. So you get the mean of the first independent variable, subtract from the original values of the independent variable, then run the model. And in so doing, you're removing the correlation. But we don't want to lose that information. So in most cases, it's better to run your multivariate regression, which will tell you where the response is. Okay. Okay, so when you analyze your data using the multiple regression, this is the equation. Here, our relationship is between volume, diameter, and height. And just similar to simple linear regression, the only thing is that now we have more than two independent variables. When you're interpreting the regression equation, you keep one independent variable constant. So we will keep height constant and then interpret diameter. So in that case, for every unit increment in diameter, keeping height constant, volume increases by 4.71 cubic meters. And when we keep diameter constant, for every unit increment in height, volume increases by 0.339. Okay, so I think we'll stop here and continue from here tomorrow. Tomorrow we'll do practicals of regression, both simple and multiple, and hopefully even model building. Sorry. So thank you so much. I am going to hand over to my colleague Thomas. This time is my boss. He has been saying I'm the boss, now is the boss. So thank you, participants. You've been so good. The questions are so well framed and informative. And we see you tomorrow. Thank you, thank you.
Yes, thank you very much, my boss, for the good work, and Ellen for answering all the questions. Yeah, I think it has been a wonderful day. We just have a bit of the issue of time here and there because 4 hours
or 3 hours a day is not that adequate, but we will always be available. If any one of you thinks you need us to come and train you, wherever you are, physically, we are more than willing to come and maybe the center will also organize another physical training in Mozambique. We'll be there. So giving you back to the forum team. Thank you.
Thank you very much, Thomas and Prof. Susan Balaba. I would like to thank also the funders for the training, the center and the World Bank, and also would like to encourage the learners to always visit the YouTube link in case there is something you want to refresh or you missed. So you can go back to the YouTube link and follow the training. And also, in case you missed the material, we always share the links for the drive where all the materials are kept and the data. So we'd like to thank also the participants that without you, it would not be a training. So we thank you very much, and we thank also the facilitators plus the funders. So we see you again tomorrow. Cheers.